The overall aim of this experiment is to isolate root tissue samples from plant seedlings grown in 96 well plates for DNA extraction and genotyping. This is accomplished by first germinating seedlings in special 96 well plates that contain agar growth media and have holes in the bottom of each well. The second step is to stack the seedling plate on top of a standard 96 well plate that is filled with water. Once the roots have grown down into the lower 96 well plate, the stacked plates are placed in a freezing block to freeze the water in the lower plate. The final step is to peel the upper plate away from the lower plate, breaking off the roots, which are frozen in the lower plate. Ultimately, the resulting root tissue samples can be used to extract DNA samples from each individual plant for genotype analysis.
The main advantage of IceCap compared to alternative methods is that it lets you isolate DNA from thousands of plants in a single day. This level of productivity is possible because with IceCap, plants are handled 96 at a time rather than the traditional 1 at a time.
Some examples of how IceCap can be used include screening large populations of plants to identify rare recombinants between closely linked loci, generating plants that carry a large number of different mapped mutations, and performing marker-assisted selection in crop species. Basically, any experiment that calls for genotyping large numbers of individual plants can potentially benefit from the IceCap method.
We originally developed IceCap for use with Arabidopsis, but we have also had success using it to collect tissue from rice and tomato seedlings. We expect that IceCap will also be effective on other species as long as their